transport is happening. So this is what we call a plasma membrane, or we can anonymously also call it a cell membrane. The basic composition of this membrane is that we have around 42% lipids or phospholipids with their polar heads and non-polar tails. Then we have about 3% carbohydrates, which can be glycoproteins or glycolipids. When attached to proteins, they are glycoproteins, and when attached to lipids, they are glycolipids. Then we have nearly 55% proteins, which can be integral or peripheral, based on whether they transverse the membrane or not. The integral proteins will transverse the membrane. For example, this serpentine protein, which transverses the membrane 7 times. Then we have peripheral proteins, which lie on either side of the cell membrane on their surface. One such example is also GPI-linked protein, which lies on the surface via GPI links. Then there are also other components which I said GPI-linked proteins. Then we have junctions like gap junctions. Macular adhesion is also known as desmosomes. And there are cell adhesion molecules which help in adhering the cell to other cell adjacent cells and with the extracellular matrix. These are further types like integrins, selectins, cadherins, and IgG family. But let's not delve into that part because our main topic is passive transport. So now, as we know that the backbone is of lipid, so it is quite obvious that lipid soluble substances will definitely have an edge over lipid insoluble substances when we're talking about transport across the membrane. Now, what is passive transport? When we define passive transport, it is simply the movement of a substance from their own higher concentration to lower concentration without the use of energy obtained from A to B hydrogen. But transport is occurring, so some kind of energy will be utilized. And this energy is the kinetic energy of the molecule itself. Then we can classify the passive transport into 3 types like diffusion, osmosis, and filtration. Furthermore, based on the nature of solute and the size of the solute, we can further divide diffusion into simple and facilitated diffusion. First, we'll talk about simple diffusion. In this, the size of particle is quite small. And secondly, the nature is also lipid soluble. So these kind of solutes will not have much difficulty to cross the membrane parts, like gases, oxygen, carbon dioxide, and other substances like urea. They follow simple diffusion to be transported across the membrane. Apart from that, this is the only kind of transport which does not require any kind of carrier or a channel. Then we have facilitated diffusion for substances which are quite larger in size or which have a polar nature. That is, they are lipid insoluble. Sometimes they can also be water soluble because of being polar in nature. Now these kind of solutes require channels to be crossed for the purpose of crossing the membrane. For example, ions, sodium ion, potassium ion, or other cations and anions, they require channels for crossing the membrane. There may be leaky channels which allow continuous flow of ions from higher concentration to lower concentration again, because we're talking about passive transport. Then there may be gated channels like voltage gated channels, ligand gated channels. In case of voltage gated channels, there is a difference in the potential. There is a potential difference across the membrane because of which the transport will occur. Then, in case of ligand gated channels, an ion or a molecule or a hormone will first bind with the receptor, and then the transport will occur because the gate will open. Now let's move on to osmosis. Osmosis in simple terms is the movement of a solvent from its own higher concentration to lower concentration through a semi-permeable membrane. Another way to describe this process in terms of solution concentration is that there is movement of solvent from a less concentrated solution to a solution of greater concentration. Now I'd like to ask the audience. This is an example wherein we have taken an aqueous solution. This is the more concentrated side and this is the less concentrated side. Now I want anyone from my batch to answer where will be the movement of water, from A to B or B to A. Now that is a subjective answer that most answer, but the main concept says that the movement of water is in both directions. When we talk about osmosis, we need to specify that there is net movement from B to A. Or we can say net movement from a hypertonic side to a hypertonic side. Now, what are these hypotonic, hypertonic, and isotonic? Subjectively, we can define as a more concentrated solution is hypertonic and a less concentrated solution is hypotonic. But conceptually, it is incorrect. Objectively, when we are specifying the concentration of the solution, we need to keep a reference. For example, we take an RBC whose concentration is isotonic with 0.9% NaCl or 5% glucose solution. So I want you to answer. For example, I've kept RBCs in an aqueous solution. Firstly, this is the aqueous solution and my solute concentration may vary. If I'm considering a hypotonic case, then there will be more solute inside this RBC membrane while comparatively less solute concentration outside within the solution. So in this case, there will be movement into the RBC, the cell will swell and hence go. In case of isotonic, the concentration outside and inside the cell is equalized. The movement will still be there, both into the cell and out of the cell, but there will be no net movement. And that is also how RBCs are stabilized in the plasma, because the plasma concentration is also 0.9% NaCl or 5% glucose. Then we talk about hypertonic solutions, wherein we have kept RBCs in a solution whose concentration is greater than its own concentration. In this case, the solvent will move out, not the solvent actually, the fluid of the RBC will move out of the RBC and therefore shrinkage will occur. So if we are clear with hypertonic, hypotonic and hypertonic terms, we can move ahead to the clinical application of osmosis. In our daily life, we breathe. Everyone breathes. So in our lungs, in the alveoli, diffusion occurs. And that is because of a pressure difference. Firstly, when we talk about partial pressure of oxygen, it is 104 mmHg. While in our blood capillaries, it is 40 mmHg because the blood is deoxygenated. Here, oxygen will move from alveoli to the plasma as well as the RBC based on where, like, in RBC there will be 97% transport of oxygen and in plasma there will be 3% transport. Apart from that, in capillaries there is 45 mmHg concentration of, partial pressure of carbon dioxide, and in alveoli there is 40 mmHg of partial pressure of carbon dioxide. So again diffusion will occur and carbon dioxide will move from its higher concentration to lower concentration, and this is how carbon dioxide is removed from our system and oxygen is taken in. Then we move on to the clinical application of osmosis. Saline. हमने कितनी बार सुना है कि चक्कर खाते दे दे, डिहाइड्रेशन हो गया, बॉटल चढ़ी है ग्लूकोज की एंड ऑल. सो उसी को सलाइन बोलते हैं. नॉर्मली जो हम सलाइन चढ़ाते हैं दैट इज नॉर्मल सलाइन. The concentration is 0.9% NaCl or 5% glucose. Like we say, these are given in cases of diarrhea, excessive vomiting or loose motion, or in case of food poisoning also vomiting is observed. So whenever there is excessive dehydration, we give an isotonic saline or a normal saline. Other types of saline are hypotonic saline, which are given for cellular rehydration. That is, the cell will be rehydrated from within. But there is a risk, because if we are not monitoring the concentration, the cell may swell and burst in the case of hypotonic saline. Then we also have hypertonic saline, wherein application of hypertonic saline, which is to treat edema. For example, like cerebral edema. What happens is that fluid is accumulated in the interstitial spaces, and when we are giving a hypertonic saline, the fluid will move into the capillaries, and it will keep on moving until and unless an equilibrium is attained and this edema situation is sorted out. Now, another risk with hypertonic saline is that, again, if the concentration is not monitored, the cells may shrink. Cerebral edema, we can observe it in cases of hemorrhage, in cases of trauma. I'm sorry, not saying about hemorrhage. In cases of trauma, in cases of shock, or in cases of injury, in cases of accidents. Apart from that, we also have, like in case of bacterial infection, for example, we have the cytotoxic B cells and NK cells, which are going to insert perforant proteins into the bacterial membrane. So what are these phosphorants going to do? They're going to create pores in the bacterial cell wall and cell membrane, which will lead to the movement of water or the fluid in our body into the bacterial cell, and therefore the cell will swell and burst, and therefore the bacterial cell will die. So this is the mechanism in our defense where osmosis is taken into account. Apart from that, we also have aquaporins in our brain, which regulate the uh, osmosis and therefore homeostasis occurs. For example, aquaporin 4 is abundant in our brain. So if there is any kind of defect in aquaporin 4, it can lead to cerebral edema. Apart from that, aquaporins are also absorbed in the renal system. For example, aquaporin 1 and 2, they are going to cause holes in their channels actually. They are going to regulate the homeostasis or osmotic balance in the renal system. For example, if we need to reabsorb more water, then in that case, ADH will be ADH will be released, adenyl cyclase will be released, and then cyclic AMP will further. This whole mechanism will further lead to creation of pores in the renal system within the distal parts and the collecting duct. So from there, water absorption will occur, and therefore, as my bachelor's already said, it is a mechanism of maintaining the homeostasis. So that's all on my part.